Emergence and implications of cell theory. Study of cell or cell biology began with the discovery of cell by Robert Hooke in 1665, who reported his work in his famous publication Microphagia. He prepared and studied thin sections of cork of dead plant material under his self-made compound microscope. He observed that cork is composed of minute honeycomb-like compartments, which he termed as cells. According to Hooke, cell is an empty space bounded by thick walls. Very little information was added to this idea in the following century. The work again started in the beginning of 19th century. Lorenz Oken, a German scientist, believed that all living beings originate from or consist of vesicles or cells. Jean-Baptiste de Lamarck in 1809 expressed similar idea and said, "Nobody can have life of its own constituent parts, are not cellular tissues, or are not formed by cellular tissues." In 1831, Robert Brown reported the presence of nucleus in the cell. Due to this discovery, Hooke's idea about the cell as an empty space was changed. It was later established that cell is not an empty space. A German zoologist, Theodor Schwann, in 1839, and a German botanist, Schleiden, in 1838, working independently, came out with a theory called cell theory. They found that cell consisted of three basic parts: nucleus, the fluid or cytoplasm surrounding the nucleus, and an outer thin covering or membrane, plasma membrane. The cell wall, they said, was an additional structure, present only in the plant cell. Keeping in view the definition of the cells could be observed in plants as well as in animals. According to the cell theory, all living organisms are composed of cells and cell products. The cell theory is one of the most fundamental generalizations in biology. It has wide-ranging effects in all fields of biological sciences. After the cell theory was presented, many details of the cell were studied, as a result of which the cell theory was extended. Rudolf Virchow, a German physician, in 1855, hypothesized that new cells were formed only by the division of previously existing living cells. It was contrary to the idea of abiogenesis. Living things arise spontaneously from non-living things, one of the prevailing but controversial ideas about origin of life at that time. Louis Pasteur in 1862 supplied experimental proofs of Virchow's hypothesis by demonstrating that microorganisms or bacteria could be formed only from existing bacteria. Original cell theory and Virchow's hypothesis gave us the basis for working definition of living things. Living things are chemical organizations composed of cells and capable of reproducing themselves. August Rizman in 1880 said, "All presently living cells have a common origin because they have basic similarities in structure and molecules." It was shown that there are fundamental similarities in chemical composition, metabolic activities, and structure, although they differ in many respects. Cells are basically similar but extraordinarily versatile. Cell is not only the structural but also functional unit of living organisms. So cell theory is very important unifying concept. The salient features of cell theory in its present form are: All organisms are composed of one or more cells. All cells arise from pre-existing cells. Cell is a basic structural as well as functional unit for all organisms. A cell is a unit of structure and function of living organisms. In multicellular organisms, there is a division of labor among cells. Different cells are specialized for different functions. The function of the organism as a whole is a result of sum of activities and interactions of different cells and of different components of the cell. In animals, muscle cells contract and relax. Nerve cells transmit impulses. Gland cells secrete. Red blood cells carry oxygen. And some stomach cells secrete gastric juice. Similarly, in plants, xylem cells conduct water and mineral salts from soil to the aerial parts of plant. Phloem cells translocate food. Sclerenchymatous cells give support to the plant. Chlorenchymatous cells carry out photosynthesis. Parenchymatous cells store surplus food. And meristematic cells produce new cells for growth and development of plant. As they perform different functions, they show great variations in shape and size. Despite the structural and functional diversity, the plant cells as well as animal cells have common plan of organization. The human naked eye can differentiate between 2 points, which are at least 1 millimeter apart. This is known as resolution of the eye. This resolution can be increased with the aid of lenses. In a typical compound microscope, the resolution is 2 micrometer, which is about 500x that of naked eye. A compound microscope is a typical laboratory microscope with at least different magnification powers. The typical ocular lenses could be 5x and 10x, but others also exist. Likewise, different types of objective lenses 20x, 40x, 100x, the magnification power of microscope is determined by multiplying x values of ocular lens and x value of objective lens. Therefore, a microscope with 10x ocular lens and 40x objective lens will have 400x magnifying power. The resolution will however remain the same, which is 500x that of the naked eye. The source of illumination in such microscope is visible light. In electron microscope, the source of illumination is a beam of electrons, and the resolution of microscope ranges between 2 to 4 angstrom, which makes it 500x greater than that of compound microscope, and 250,000x greater than that of naked eye. This means that 2 points which are 2 to 4 angstrom apart can be differentiated with the help of electron microscope. The revelation of complexity of structure of various cellular organelles is closely linked with the development of microscopy and improvement in resolution power of the microscope. Structure of a generalized cell. Structure of a cell can be studied under light microscope as well as electron microscope. The modern technology enables us to isolate various components of cells, including its organelles, by a process of cell fractionation and study their structure and function in detail. During cell fractionation, the tissues are homogenized or disrupted with the special instruments, and the various parts of the cells are separated by density gradient centrifugation. This separation is achieved by spinning the homogenized or disrupted cells in a special medium, in a centrifuge at medium speed. The various cellular parts separate out in different layers depending upon their size and weight. Some cellular components require very high speed for separation from other parts of the cells. This is achieved through ultracentrifugation. A cell consists of the following basic components. Plasma membrane, also a cell wall in plant cells. Cytoplasm, containing cell organelles. Nucleus with a nuclear or chromatin material. In the traditional system of classification, all organisms are divided into plants and animals. The cells of plants and animals can be distinguished by the presence or absence of cell wall. Cells of animals and plants are complex and have a distinct nucleus. That is, the chromatin material is bounded by membrane and are called eukaryotic. On the other hand, the primitive type of cells, such as bacteria, lack a definite nucleus and are said to be prokaryotic. In prokaryotes, the nuclear material is directly submerged in the cytoplasm and is not separated from it by membranes. The eukaryotic cells vary greatly in size. They could be as big as an ostrich egg. Most of the cells are microscopic and are not visible with the naked eye. Their size is measured in micrometers. The use of the modern technology has made it possible to study the following components of the cells in details. Plasma membrane or cell membrane is the outermost boundary of the cell. However, in most plant cells is covered by cell wall. Cell membrane is chemically composed of lipids and proteins. 60 to 80% are proteins, while 20 to 40% are lipids. In addition, there is a small quantity of carbohydrates. Many biologists contributed to establish the structural organization of cell membrane. It was proposed earlier that cell membrane is composed of lipid bilayer, sandwiched between inner and outer layers of proteins. This basic structure is called unit membrane and is present in all cellular organelles. The modern technology has revealed that lipid bilayer are not sandwiched between two protein layers. The protein layers are not continuous and are not confined to the surface of the membrane, but are embedded in lipid layers in a mosaic manner. This discovery led to the proposal of fluid mosaic model. This model at present is the most accepted one. Cell membrane also contains charged pores through which movement of materials take place, both by active and passive transport. Transport of materials is one of the vital roles it plays for the cell. It offers a barrier between cell contents and their environment, allowing only selective substances to pass through it. Thus, it is known as differentially permeable or selectively permeable membrane. The substances which are lipid soluble cross it more easily than others. Therefore, it regulates the flow of materials and ions to maintain a definite gradient. Many small gas molecules, water, glucose, etc., being neutral, can easily cross, while ions, being charged particles, have some difficulty in crossing. Many substances which are not needed constantly enter the cell by passive transport. Others are taken up against the concentration gradient. That is, they move from area of low concentration to the area of high concentration. This uphill movement of materials requires energy and is termed as active transport. The energy used for this movement is provided by ATP. In many animal cells, the cell membrane helps to take in materials by enfoldings in the form of vacuoles. This type of intake is termed as endocytosis, which can be either phagocytosis to engulf solid particles or pinocytosis to take in liquid material. In neurons or nerve cells, the cell membrane transmits nerve impulses from one part of the body to other to keep coordination. The outermost boundary in most of the plant cells is cell wall. The cell wall of plant cell is different from that of prokaryotes, both in structure and chemical composition. It is secreted by the protoplasm of the cell. Its thickness varies in different cells of the plant. It is composed of three main layers: primary wall, secondary wall, and the middle lamella. The middle lamella is first to be formed in between the primary walls of the neighboring cells. The primary wall is composed of cellulose and some deposition of pectin and hemicelluloses. Cellulose molecules are arranged in a crisscross arrangement. The primary wall is a true wall and develops in newly growing cells. The secondary wall is formed on the inner surface and is comparatively thick and rigid. Chemically, it is composed of inorganic salts, silica, waxes,
cutin, lignin, etc. Prokaryotic cell wall lacks cellulose. Its strengthening material is peptidoglycan or murine. Fungal cell wall contains chitin. Cell wall is very important. It provides a definite shape to the cell and keeps it rigid. It doesn't act as a barrier to the materials passing through it.
Cytoplasm. The living content of eukaryotic cells are divided into nucleus and the cytoplasm. The two collectively form protoplasm. Cytoplasm consists of an aqueous ground substance containing a variety of cell organelles and other inclusions such as insoluble waste and storage products. The soluble part of cytoplasm is called cytosol. It forms the ground substance of the cytoplasm. Chemically, it is about 90% of water. It forms a solution containing all the fundamental molecules of life. In the cytosol, small molecules and ions may form true solutions, and some large molecules form colloidal solutions. Colloidal solutions may be a sol, non-viscous, or a gel, viscous. Peripheral parts of cells are often like a gel. The most important function of cytoplasm is to act as a storehouse of vital chemicals. It's also a site of certain metabolic processes such as glycolysis. In living cells, the cytoplasm contains several cell organelles such as endoplasmic reticulum, mitochondria, Golgi complex, nucleus, plastids, ribosomes, lysosomes, and centrioles. The free-floating cell organelles like mitochondria move about in cytoplasm due to cytoplasmic streaming movements. This is an active mass movement of the cytoplasm. Endoplasmic reticulum. Under an electron microscope, a network of channels is seen, extending throughout the cytoplasm. These channels are often continuous with the plasma membrane and also appear to be in contact with the nuclear membrane. This entire system of channels is the endoplasmic reticulum. These membranes vary widely in appearance from cell to cell. The materials present in these channels is separated from the cytoplasmic materials by the spherical or tubular membranes called cisternae. There are two morphological forms of endoplasmic reticulum: a rough form with attached ribosomes and a smooth form without ribosomes. The rough surfaced endoplasmic reticulum is involved in synthesis of proteins. After synthesis, the proteins are either stored in the cytoplasm or exported out of the cells through these channels. The smooth-surfaced endoplasmic reticulum helps in metabolism of a number of different types of molecules, particularly lipids. They also help to detoxify the harmful drugs. In some cells, smooth endoplasmic reticulum is responsible for transmission of impulses. For example, muscle cells and nerve cells. In addition, smooth endoplasmic reticulum also plays an important role in the transport of materials from one part of the cell to the other. Endoplasmic reticulum also provides mechanical support to the cell so that its shape is maintained. Ribosomes. Cells contain many tiny granular structures known as ribosomes. Palade was the first person to study them. Eukaryotic ribosomes are composed of an equal amount of RNA and proteins. Hence, they are ribonucleoprotein particles. The RNA present in the ribosomes is called ribosomal RNA. Ribosomes exist in two forms, either freely dispersed in cytoplasm or attached with rough endoplasmic reticulum as tiny granules. Each eukaryotic ribosome consists of two subunits. The larger subunit sediments at 60S, while smaller subunit sediments at 40S. Two subunits on attachment with each other form an 80S particle. This attachment is controlled in the presence of magnesium ions. The ribosomes are attached to the messenger RNA through small ribosomal subunit. A group of ribosomes attached to messenger RNA is known as polysome. New ribosomes are assembled in the nucleolus of the nucleus, from where they are transported to the cytoplasm via the pores in the nuclear membrane. The factory of the ribosomes is nucleolus, while that of protein synthesis is ribosomes. Golgi apparatus. Golgi apparatus was first discovered by Golgi in 1898. This apparatus, which was found virtually in all eukaryotic cells, consists of stacks of flattened membrane-bound sacs called cisternae. These cisternae, together with associated vesicles, are called Golgi complex. It's a complex system of interconnected tubules around the central stacks. Cisternae stacks are continuously formed by fusion of vesicles, which are probably derived by budding of smooth endoplasmic reticulum. Their outer convex surface is the forming phase, while the inner concave surface is the maturing phase. The whole stack consists of a number of cisternae, thought to be moving from the outer to the inner phase. Transport vesicles are pinched off at the maturation phase of Golgi apparatus. The arrows show the direction of flow of protein products synthesized on the ribosomes. These proteins are converted into glycoproteins in the Golgi apparatus. Golgi complex is concerned with the cell secretions. Secretions are the products formed within the cell on the ribosomes and are then passed to the outside through endoplasmic reticulum and Golgi apparatus. The secretions are converted into the finished products and are packed inside the membranes before export. For example, in mammals, The pancreas secretes granules containing enzymes that help in digestion. The Golgi complex has a role of formation of these granules. The proteins or enzymes which have to be transported out of the cell pass through the Golgi apparatus. The most important function of this apparatus is to modify the proteins and lipids by adding carbohydrates and converting them into glycoproteins or glycolipids. Lysosomes. Lysosomes are cytoplasmic organelles and are different from others due to their morphology. These were isolated as separate components from first time by D. Dewey in 1949. Lysosomes. Lyso means splitting and soma means body, are found in most eukaryotic cells. Any foreign object that gains entry into the cell is immediately engulfed by the lysosomes and is completely broken into simple digestible pieces. The process is known as phagocytosis, or eating process of a cell. They are most abundant in those animal cells which exhibit phagocytic activity. They are bounded by single membrane and are simple sacs rich in acid phosphatases and several other hydrolytic enzymes. These enzymes are synthesized on rough endoplasmic reticulum and are further processed in the Golgi apparatus. The processed enzymes are budded off as the Golgi vesicles and are called primary lysosomes. Lysosomes contain those enzymes which can digest the phagocytosed food particles. Sometimes under abnormal circumstances, like starvation or as normal physiological process, the parts of the cell are engulfed by the primary lysosomes and are digested to generate energy. The lysosomes which eat parts of their own cells are known as autophagosomes. The digestive vacuoles and autophagosomes are known as secondary lysosomes. They are also involved in autophagy or self-eating. During this process, some old, worn-out parts of the cell, such as old mitochondria, are digested. In this way, materials of the cell may be recycled and cell may be renewed. Their enzymes can also result in degeneration of cell, as may occur due to some developmental processes. Lysosomes also release enzymes for extracellular digestion. Several congenital diseases have been found to be due to accumulation within the cell of substances such as glycogen or various glycolipids. These are also called storage diseases and are produced by mutation that affect one of the lysosomal enzymes involved in catabolism of certain substances. For example, in glycogenosis type 2 disease, the liver and muscle appear filled with the glycogen within membrane-bound organelles. In this disease, the enzyme that degrades glycogen or glucose is absent. About 20 such diseases are known these days, which are because of absence of particular enzyme. For example, Tay-Sachs disease is because of absence of enzyme that is involved in catabolism of lipids. Accumulation of lipids in brain cells lead to mental retardation and even death. Peroxisomes. Did and co-workers isolated in 1965 particles from the liver cells and other tissues, which were enriched with some oxidative enzymes such as peroxidase, catalase, glycolic acid oxidase, and some other enzymes. The name peroxisome was applied because this organelle is specifically involved in the formation and decomposition of hydrogen peroxide in the cell. These are single membrane enclosed cytoplasmic organelles found both in animals and plant cells. These are characterized by containing hydrogen peroxide producing oxidases and catalases. They are approximately 0.5 micrometer in diameter. They have also been found in protozoa, yeast, and many other types of the cells in higher plants. Glyoxysomes. Plants contain an organelle which, in addition to glycolic acid oxidase and catalase, also possess a number of enzymes that are not found in animal cells. These organelles, called glyoxysomes, are most abundant in plant seedlings, which rely upon stored fatty acids to provide them with the energy and material to begin the formation of a new plant. One of the primary activities in these germinating seedlings is the conversion of stored fatty acids to carbohydrates. This is achieved by the cycle glyoxylate cycle, the enzymes of which are located in glyoxysomes. In plants, peroxisomes play an important role in both catabolic and anabolic pathways. In seeds rich in lipids such as castor beans and soybeans, glyoxysomes are the sites for breakdown of fatty acids to succinate. This organelle is present only during a short period in the germination of the lipid-rich seed and is absent in lipid-poor seeds such as pea. Vacuoles. Although vacuoles are present both in animal and plant cells, they are particularly large and abundant in plant cells, often occupying a major portion of the cell volume and forcing the remaining intracellular structures into a thin peripheral layer. These vacuoles are bounded by a single membrane and are formed by coalescence of smaller vacuoles during plant's growth and development. Vacuoles serve to expand the plant cell without diluting its cytoplasm and also function as site for storage of water and cell products or metabolic intermediates. The plant vacuole is a major contributor to the turgor that provides support for the individual plant cell and contributes to rigidity of the leaves and younger parts of the plants. Cytoskeleton. Cytosol contains cytoskeletal fabric formed of microtubules, microfilaments, and intermediate filaments. The main proteins that are present in cytoskeleton are tubulin in microtubules, actin, myosin, tropomyosin, and others, which are also found in muscles. Several cell organelles are derived from special assemblies of microtubules, for example, cilia, flagella, basal bodies, and centrioles. The movement of cycloses and amoeboid movement are because of microfilaments, whereas intermediate filaments are involved in determination of cell shape and integration of cellular compartments. Microtubules are long, unbranched, cylinder tubulin protein structures. One very important function of microtubules is their role in assembly and disassembly of spindle structure during mitosis. Microfilaments are considerably more slender cylinders made up of contractile actin protein linked to the inner face of plasma membrane. They are involved in internal cell motion. Intermediate filaments have a diameter in between those of microtubules and microfilaments. They play a role in maintenance of cell shape. Centriole. Animal cells and cells of some microorganisms and lower plants contain two centrioles located near the exterior surface of the nucleus. In cross section, each centriole consists of a cylindrical array of 9 microtubules. However, each of the 9 microtubules is further composed of 3 tubules. The two centrioles are usually placed at right angle to each other. Just before a cell divides, its centrioles duplicate, and one pair migrates to opposite sides of the nucleus. The spindle then forms between them. They are absent in higher plants. Centrioles play an important role in location of furrowing, during cell division and in the formation of cilia. Mitochondria. Mitochondria are very important organelles of eukaryotic cells because they are involved in manufacture and supply of energy to the cell. They are also known as powerhouses of cell. Under compound microscope, they appear to be vesicles, rods, or filaments. Under electron microscope, they show complex morphology. Although their number, shape, and internal structure vary widely, a mitochondrion is bound by two membranes. The outer membrane is smooth, while the inner membrane forms infoldings into inner chamber, called mitochondrial matrix. These infolds are called cristae. The mitochondrial membranes are similar in structure to other cell membranes. Detailed studies have shown that mitochondria also contain DNA, as well as ribosomes. The presence of ribosomes in DNA indicates that some proteins are synthesized in them. It is a self-replicating organelle. The inner surface of cristae in the mitochondrial matrix has smaller knob-like structures known as F1 particles. Mitochondrial matrix contain in it large number of enzymes, coenzymes, and organic and inorganic salts, which help in several vital metabolic processes like Krebs cycle, aerobic respiration, fatty acid metabolism, and many more. As a result of these metabolic processes, the energy extracted from the organic food is transformed into energy-rich compound ATP or adenosine triphosphate, and the ATP then provides energy to the cell on demand. The size and number of mitochondria varies and depends on physiological activity of the cell. Mitochondria extract energy from different components of food and convert it in the form of ATP. This energy is used for various cellular activities. The spent energy, which is in the form of ADP, is regenerated by mitochondria into ATP. Mitochondria is therefore described as powerhouse of the cell. Plastids. Membrane-bound, mostly pigment-containing bodies present in the cell are called plastids. Plastids are present in plant cells only. There are three main types of plastids: chloroplast, chromoplast, leucoplast. In photosynthetic plant cells, there are membrane-bound structures containing a green pigment called chloroplast. The green pigment is an organic compound, chlorophyll, which helps the cell to absorb light energy and utilize it to manufacture food. Chlorophyll molecules resemble the heme group of hemoglobin, a protein that is used to transport oxygen. The main difference between these two molecules is that chlorophyll has magnesium ions, while heme has iron as the central atom. Chloroplasts vary in their shape and size, with a diameter of about 4 to 6 micrometer. Under light microscope, they appear to be heterogeneous structures, with small granules known as grana, embedded in the matrix. Under electron microscope, a chloroplast shows three main components: the envelope, the stroma, and the thylakoid. The envelope is formed by a double membrane, while stroma covers most of the volume of chloroplast. Stroma is a fluid which surrounds the thylakoids. It contains proteins, some ribosomes, and a small circular DNA. It is in this part of chloroplast where carbon dioxide is fixed to manufacture sugars. Some proteins are also synthesized in this part. Thylakoids are the flattened vesicles which are arranged themselves to form grana and intergrana. A granum appears to be a pile of thylakoids, stacked on each other like coins. On an average, there are 50 or more thylakoids piled to form one granum. On the layers of thylakoids, chlorophyll molecules are arranged, and that's why granum appears to be green. Each granum is interconnected with others by the non-green part called intergranum. Membranes of grana are the sites where sunlight energy is trapped and where ATP is formed. Chloroplasts are self-replicating organelles. Chromoplasts. They impart colors to the plants other than green. They are present in petals of the flowers and in ripened fruits. They help in pollination and dispersal of seeds. Leucoplasts. They are colorless. They are triangular, tubular, or of some other shape. They are found in underground parts of the plants and store food.
Presence of cell nucleus was reported in 1831 by Robert Brown. Its early discovery was undoubtedly due to its prominence in many cells, where it stands out slightly darker than the surrounding cytoplasm. It controls the life and activities of cell. In animal cells, it generally occupies the central space, while in the case of plant cells, it's pushed towards periphery due to presence of a large vacuole. Nucleus may be irregular or spherical in shape. Generally, the cells have one nucleus and are called mononucleate. On the other hand, the cells with the two nuclei are binucleate and with more than two are multinucleate. Nucleus is only visible when the cell is in non-dividing stage. It contains chromatin network and soluble sap-like nucleoplasm. In dividing cells, the nucleus disappears, and the chromatin material in it is replaced by the chromosomes. The heredity material is in the form of chromosomes, which control all the activities of cell. DNA, RNA, and proteins, including enzymes, form the chemical composition of the nucleus. Nucleus consists of nuclear membrane, nucleoli, nucleoplasm, and chromosomes. Nucleus is surrounded by nuclear membrane, which separates the nuclear material from the cytoplasm. The nuclear membrane is actually the nuclear envelope composed of two membranes. The outer membrane is at places continuous with the endoplasmic reticulum, while the inner membrane encloses the nuclear content. The outer and inner membranes are continuous at certain points, resulting in the formation of pores, the nucleopores. The nucleopores allow the exchange of materials between the nucleus and cytoplasm. The number of nucleopores is highly variable. The undifferentiated cells, such as eggs, have numerous pores, about 30,000 per nucleus, whereas differentiated cells, such as erythrocytes, have only 3 to 4 pores per nucleus. Each pore has a definite structure which controls the traffic of substances passing through them. Nucleolus. It's a darkly stained body without any membranous boundary to separate it from the rest of the nuclear material. There may be one or more nucleoli in the nucleus. The ribosomal RNA is synthesized and stored in it. It's composed of two regions, the peripheral granular area composed of precursors of ribosomal subunits, and a central fibrillar area consisting of large molecular weight DNA and RNA. It is the nucleolus where ribosomes are assembled and then exported to the cytoplasm via nucleopores. Chromosomes. Nucleus is often deeply stained with the basic dye because of the chromatin material. During cell division, chromatin material is converted into darkly stained thread-like structures known as chromosomes. Under a compound microscope, chromosomes appear to be made of arms and centromeres. Centromere is a place on chromosome where spindle fibers are attached during cell division. Each chromosome consists of two identical chromatids at the beginning of cell division. Chromatid is the exact replica of chromosome, which are held together at centromere. A chromosome is composed of DNA and proteins. All the information necessary to control the activities of cell is located on chromosomes in the form of genes, which are transferred from one generation to the other. The number of chromosomes in all individuals of the same species remain constant generation after generation. In men, each cell contains 46 chromosomes. There are 8 chromosomes in fruit fly, Drosophila melanogaster, 16 chromosomes in onion, 48 in potato, and 14 in garden pea. The number of chromosomes in normal body cells is diploid, 2n, whereas haploid chromosome number or n is present in germ cells. Like human sperms and eggs have 23, while those of Drosophila has 4 chromosomes. Prokaryotic and eukaryotic cell. Biologists have divided cells into two types, prokaryotic and eukaryotic. The differences between these two types of cells are mainly based upon the structure of their nuclei. Eukaryotes have a very well-defined nucleus in which nuclear material, chromosomes or DNA, is enclosed in double nuclear membrane. In prokaryotic cells, however, genetic material or DNA is without any nuclear membrane covering and is directly submerged in the cytoplasm. Organisms possessing prokaryotic cells are called prokaryotes, and those possessing eukaryotic cells are called eukaryotes. Prokaryotes include bacteria and blue-green algae. Eukaryotes include all other unicellular or multicellular organisms, such as animals, plants, fungi, and protista. Prokaryotic cells generally lack many of the membrane-bounded structures found in eukaryotic cells. For example, mitochondria, endoplasmic reticulum, chloroplast, and Golgi apparatus are absent in prokaryotic cells. Since there is no nuclear membrane, a prokaryotic cell has no distinct nucleus and its DNA molecule is directly suspended in the cytoplasm. Prokaryotes have small-sized ribosomes of 70s compared to eukaryotes of 80s. In prokaryotes, mitosis is missing and the cell divides by binary fission. Because of their simpler structure, it was widely accepted for a long time that prokaryotic cells represent a more primitive stage of evolution than eukaryotic cells. Perhaps the most distinctive feature of prokaryotic cell is its cell wall, bound covalently to shorter chains of amino acids forming peptidoglycan or murine. The entire cell wall is often regarded as a single huge molecule or molecular complex called nucleus. The cell wall of plants is generally made up of cellulose and is differently structured than that of a bacterium.